Today's pre-lab will be on meiosis. Last week we worked with what we refer to as a mitosis. Mitosis is a process by which we take one cell that would have, say, a full complement of chromosomes. Say for a human being would have 46 chromosomes. Actually, that is 23 pair. 2 number 1s, 2 number 2s, 2 number 3s, and so forth. 2 number 23s. And if you add that up, that's a total of 46. And when the cell finishes its cell division, you end up with 2 cells that will have the exact same 46 chromosomes. So the quantity of chromosomes quantitative and qualitative nuclear division would be amitosis. Now, in a meiosis, this is a process by which we create gametes. So, of course, at some point we enter puberty, and during that period of time we produce gametes. So, of course, the female gonad, which is a structure that produces the ovum or the egg, and the egg is the gamete, and that, of course, begins at puberty. And in males, the testes, which would be the male male gonad, produces the gamete, which, of course, would be the sperm. So that, again, begins at puberty. So what we want to do here is take the full genetic complement, which for a human being, the full genetic complement would be the 46 chromosomes. So we have a total of 46. And in a meiosis, it's actually a two-stage cell division. You take one cell and you divide it into 2 cells, and then you take each one of those and you divide those a second time. So you have 2 cell divisions, and when you're finished with the 2 cell divisions, you actually end up getting 4 cells, and each of the 4 cells will only have 1/2 of the full genetic complement. So in this case, if your full genetic complement is 46 chromosomes, then 1/2 of that would be 23 chromosomes. So the full genetic complement, they will actually tell us, is a 2N, and that's 2 of each type chromosome. Again, 2 number 1s, 2 number 2s, 2 number 3s. So our diploid, you get 2 of each chromosome. That's diploidy, so two copies. The full diploid state for a human would be 46. And then just the N, just the single haploid state, which is what they call it, the haploid, you only get 1 copy of each chromosome. So 1 number 1, 1 number 2, 1 number 3, and 1 number 4, so forth. So the ovum, or again the egg, as we sometimes refer to it, will only carry 23 chromosomes, again that being the haploid gamete for the female, and the sperm, which of course also carries the haploid state, which is a 23, and that is the male gamete, and that would be 1 number 1, 1 number 2. The sperm and the egg, you end up getting this cell, which is a diploid cell, which we refer to as a zygote, and that has the full genetic complement, the 46 chromosomes. So once again, we reestablish the full diploid state, which of course is then the 46, and that of course is the diploid. At that point, you really begin to go through mitosis. So you just beginning to, you start the process where you take 1 cell with 46, you divide it into 2 cells that will have 46 chromosomes. So we end up, of course, once again with the diploid state in both cells. So you go from 1 to 2 cells, 2 to 4, 4 to 8, until eventually at some point you're at full maturation, fully mature human being. And then again, so this is the zygote, and at some point the process of puberty begins, and you begin to go through meiosis once again to produce the gametes. So it's kind of the life cycle of the human being, the entire life cycle. Now let's go back to meiosis. The first cell division that we have, going from the 46 chromosomes to 2 cells, this is what we refer to as meiosis 1, the first cell division, meiosis 1, and then the second cell division, so this is also a meiosis 1 here, and the second cell division would be a meiosis 2, second cell division. All right, so we've reduced our chromosome number exactly in half from 46 to 20 to 23. Right, so this is, again, reductional division if we're talking about a meiosis. If it's a mitosis, you do not reduce the chromosome number. All right, so we go from diploid to haploid. And if you are looking at, say, a cell that is diploid, having both number 1 chromosomes and 2 number 2 chromosomes and 2 number 3 chromosomes, let's say these are both number 1 chromosomes. One originally comes from the father and one comes from the mother. The pair of these number 1 chromosomes, these are referred to as homologous chromosomes. So they're the same length, same size, they carry the same genetic traits. That being, say, eye color, hair color, and a number of other traits that would be possible. So though that information would be carried at a specific place on those homologous chromosomes. Let's say at this particular section we have a code for eye color. A capital B would be a code for brown eyes. A small b might be the code for blue eyes. So in this case we have 2 different forms of a gene, and that's exactly what we're working with. It's just a little section of the chromosome which would be the gene. So the actual location of that gene is what they call a locus. So that is just location of a gene, where you find it on the chromosome. It could have been found at this spot. It may have been found a little lower on the chromosome, but that is the location. And then location of a gene. A gene just codes for a specific protein, in this case a brown pigment, or actually blue pigment is an absence of pigment, and we'll discuss that. So gene is a genetic code for a protein. And then you'll notice that there may be 2 different forms of a gene. Like in this particular example, we have a gene for brown eyes and a gene for blue eyes. So you do get 2 possible different genes. Those are referred to as the alleles, and the alleles are just the different forms of a gene. Now, it is possible that both of the alleles could be identical. Say, for example, you could have received a capital B gene from the mother and a capital B gene from the father. In that case, these are called homozygous alleles. And because they're both dominant, they refer to this as homozygous dominant. Both being identical and both of those being dominant genes, brown being a dominant gene to blue. And then, of course, you could have homozygous recessive. So if you receive the gene for blue eye color from the mother and a gene for blue eye color from the father, both of those are identical, and so they're homozygous, but they are different in that they are, instead of the dominant form, they are both the recessive form, so they're homozygous, but they are the recessive condition. All right, and then of course, we had the actual two different forms of the gene. Technically, those would be the alleles, but this would be what we refer to heterozygous. So one is dominant, one is recessive, so they're certainly not identical. One is dominant, one is recessive, and that would be what we call hetero. Of course, hetero giving us the suffix, which would mean different. Homo being the suffix for identical. And so these are homozygous recessive genes. These are heterozygous, and we don't have to use recessive or dominant because one is dominant and then the other one is actually recessive. So that is not necessary that you put in, I guess you would have to, if you wanted to use that, you could say heterozygous, dominant, and recessive. But of course that's not used. You just say heterozygous. That automatically tells you that they are both going to be different. All right, that's just a little bit of terminology to go with this information. We will, in lab, go through and talk about the different stages. So we do have a meiosis 1 and a meiosis 2, two cell divisions. So really, for meiosis 1, the first division, meiosis 1, you do have a prophase 1, a metaphase 1. So let's talk about meiosis 1. Again, a prophase 1, a metaphase. I don't usually talk about prometaphase. We did have a prometaphase in mitosis. They don't typically use that when you're talking about a meiosis. So all of this is just the two cell divisions that you do have occurring in meiosis. So a prophase 1, a metaphase 1, a anaphase, and anaphase 1, telophase 1, and then you also have the second cell division, which would be meiosis 2. So let's go ahead and put the telophase 1 in here. All right, so on the second cell division, we have the meiosis 2, the second cell division. So again, we have a prophase 1, so this would be prophase 2, second, and then this would be a metaphase 2, and anaphase 2, and then a telophase 2. So just to kind of review that again, we started with a cell that had 46 chromosomes. We went through a meiosis 1. We ended up getting two cells, one half the original size. So we ended up doing the prophase 1, metaphase 1, anaphase 1, phase 1. And then we took each one of these and we went into then the meiosis 2, the second cell division. So all of these will be meiosis 2. And so then we do a prophase 2, metaphase 2, anaphase 2, telophase 2. And when you're finished with that second cell division, we have four cells, each with 23 chromosomes, or that would be the haploid condition of this particular cell, which of course this is a particular cell. This could either produce the egg, which we refer to as the ovum scientifically, or it could produce the sperm, which scientifically we refer to as the sperm. So two cell divisions get to the haploid state. All right, so from the diploid chromosome number, because if we didn't do that, we'd have 46 chromosomes in an egg and 46 chromosomes in a sperm. And that being the case, when those united to create the zygote, you would really end up getting a total of 92 chromosomes in the zygote, and of course that means that the organism, which in humans really never would develop, but in plants it does. There is a state in which you actually get, instead of just two number one chromosomes, you would actually get four number one chromosomes. It's called a tetraploid, so that's a little different situation. We should have time to discuss that. All right. And meiosis in males, they refer to as a spermatogenesis. That's just the process of producing sperm, spermatogenesis. Just as in the Bible, a genesis is a creation of sperm, spermatogenesis. For the production of the egg, we'll call it an oogenesis. Once again, a genesis being a creation, in this case a creation of the egg. And so those are the two meiotic divisions, and we'll discuss those again as we go through the process of lab. And you will be able to see a microscope slide of the ovum, which is a cross-section of the ovary, and depending on what cross-section we actually look at, you may see the egg, or you may see a precursor to the egg, and then also you'll see a cross-section of a testis, and you should be able to see some sperm or the development of the sperm in the testis. Okay, so that will be a pre-lab for the meiosis, and we will be doing that. And that should be all we'll need to take care of for this particular lab preview. Okay, so trying to get my cursor back. There we go. I think that should do it. All right.